Okay, well, hello and welcome to today's webinar. My name is Melissa. I'm a Scientific Training Officer in the training team at the EBI, and we organise a series of webinars like this that run every 2 weeks. I'm going to hand over now to Reza, who's going to tell us a little bit more about metabolomics and how you can use it in biological research.
All right. I hope you can hear me now. Sounds is good. I'll try to go a bit slower at the beginning because I know many people are joining now. This is more giving you a background in metabolomics. This is mostly main and thought of about people who want to know more about metabolomics, not just those who are doing metabolomics. So I know some of you, a few of you, Iswa, I know you're there, you're an expert, are there listening to the same webinar. But so this will be quite basic, intended for people who really want to know more about metabolomics. I'll try not to make it too boring, so at least it will be generic interest if you even do or starting to work in metabolomics. All right, just some definitions and background in case. There are several different words that use for metabolomics that describe more or less the same thing. One of the key words probably are called metabolome. So what is a metabolome? This is the sort of total number of metabolites that you can find in a cell, organism, or tissue. There are other names that did also describe the similar things. For example, studying the metabolites and metabolome is usually called metabolomics. That they use some sort of analytical techniques, some sort of extractions, combination of different techniques to be able to study range of different molecules and metabolites, and how they affect and interact with the biological or living system, how they interact with the environment, how these things are changed effects during the time. There are also other names which refer to the same thing. For example, metabolomic also has been used in parallel to metabolomics. So the one below, there's a trend lines of Google search based on keywords, and metabolomics still is the most used keyword by Google Analytics. Has been the keyword looking for metabolomics. And other terms such as metabolomic, once they were high, the numbers are going down. There are other key terms used, profiling, metabolic profiling methods. Also, there are anonymous to a similar studying of metabolome or metabolites. There are also other terms, for example, phenome. Phenome referring to studying the type of phenotypic outcome of these metabolic events. So these are all names, but all referring to same and similar science of studying small molecules. So I slowly try to say what are small molecules and what we mean, but for that there is a bit of a background. Obviously, they are quite familiar. Most of you are in some sort of a biological settings and study, and you are quite familiar with these slides, but I go through it anyway to make some points. The points that everyone knows about the reductionist system, that somehow there are genes, that genes translate into some sort of protein enzyme, and these are the molecules that interact with the environment and change metabolome. And we are already living in a chemical soup, and our environment is full of nutrients that we require, oxygen, and other molecules that are required for our living and system to continue. And these are the small molecules metabolite interacting via a proteome system to be able to regulate and change and do the changes. However, this reductionist system is not very well describe everything that we could see, because the whole biology doesn't care for our classification. They really have a serving function, something that comes via evolution and has a meaning and purpose for a living organism. So these things are, rather than being a reductionist, they're now more perceived to be integrative, meaning that all systems combine through loopbacks, through feedbacks, that your metabolites can regulate your protein, or proteins can regulate some sort of transcriptomes, and these changes reflect back how active a gene can be or cannot be. So these things can be done in a more integrative way. I mean, the best examples that people giving studying of a system biology is silkworm metamorphosis. So everyone is familiar that both how a silkworm goes into different phases of metamorphosis, but all of the genes and genomes of this species is quite well known. But just knowing the gene doesn't describe how the silkworm change and goes into metamorphosis. So it's a complex mixture of different things that the gene interact via different systems of proteome, metabolome, transcriptome, others, that they end up having a trait, a phenome, a feature, required of a living organism to change and evolve and adapt. More now about metabolomics. I guess here I want just to illustrate that when you talk about metabolism or metabolomics or metabolite, this is what people usually see, a sort of chemical and chemistry reactions that are well defined, follow certain chemical rules that are networked and adjusted. And this is true, so this is correct, but it's not the entire picture. As I said before, a lot of these changes and traits in metabolism are not independent. They are related to the lifestyle and choice of diet, to whether how much you exercise, what do you eat, and also which environment do you live on. And they're not interact or separated from how genes you inherit and how they react and translate into proteome or your enzymes, and how they feedback via different loops to different type of cells and organisms that have to live, organs that have a harmonical way of functioning with one another, communicating, being able to provide ability to live and survive and to progress. Some timelines varies again. Translation between these phenomena of different omics are quite difficult, and timescales are not very well known. It's of quite interest, a lot of people trying to study and understand how metabolome, how data from other omics can be translated, linked, fused to one another. Main issue is timelines. Metabolites usually are quite fast and responsive. So changes in metabolome is quite responsive because an organism has to respond to changing environment quite quickly. So if these are delayed, the survival rate goes down. While something changes in genomes or respond from genome is quite slow. You don't need to quickly respond to a toxin. You accumulate it, and these have the long-term consequences. In between those, the transcriptome and proteome have also a variety of responses that need to be quickly because maybe they're related to some enzyme or some sort of activity related to metabolite that need to be quite fast and responsive, or scaling something down or reducing, and changing to a response. But these things vary quite a lot. So not all the systems are a linear response. It changes and varies depending on which type of protein, transcriptome, or which system of metabolome we under investigating and we are looking at. So what people do? People usually we like as to classify things, to profile stuff, to put them into categories. And this is what a lot of metabolomics people do. They profile stuff. Metabolic profiling is also very well used. And this is not something new, as I want to describe from a urine wheel or uroscopy that can date back to ancient times, from Egyptian, Babylonian, Indian, and all very well documented in Byzantine medicine that they classified different type of urine. So the analytical instrument at the time was smell, sight, taste, to be able to see what different type of colors, smells, or looks, or textures of urine vary. And then they try to put some sort of meaning. So people with certain color, if they're red in the urine, they might have certain symptoms related to the disease they have. So they start to profile based on urine profiling. So as you can see, urine profiling for metabolomics is, as a concept, is another new concept, but it's an ancient system. And one of the oldest metabolites that probably was discovered and very known from ancient time is glucose. So sugary taste or sweet taste of urine always was a good indicative for people that there is something not right. So they might not call it diabetes, or they might not know what happened, but they knew the outcome and consequence of high sugar or sugary or sweet urine. This is usually happens in people with type 2 diabetes. So not discovering a biomarker, it happened at the time, and probably glucose is one of the oldest discovered biomarkers that is a metabolite. Still to this date, it's the most common use for monitoring as well. So finding a biomarker, the useful of it is to be able to trace, to monitor, to track a disease. And still, people with type 2 diabetes, finger prick tests is the most common way of measuring blood glucose. But these are improving substantially by advent of technology, by usage of technology, by being able to now continuously monitor using everyday devices. So this is the area that I quite like, and I quite like this example. That if you do, for example, just finger prick test, this is sort of a line here by red dots on the graph on top left. You seem, and the blue zone is the target normal range zone. You might seem that, oh, everything's fine. I mean, my glucose levels are not going up and down. It seems to be all in the target range. It is once you be able to continuously monitor CGM, continuous glucose monitoring, that you will see, oh, okay, it's a lot of more changes, huge changes that went unnoticed because I have no idea about change, trends, and directions that this glucose was taking. And this gives you far more insight because it's not just the carbohydrate you have for people with type 2 diabetes that they need to adjust. Soon they realize it's their lifestyle, the choice of exercise, the hormones affect, the growth hormones that they might have at different young age, old age, changing in the environment, stress, all of these things are actually a factor in changing metabolome. And this is what's happening to all of us. The difference between us will be that there is a pancreas that manages automatically, but they need to do it manually. Once you have to do manually, really realize that how many factors are there to factor in. And it's overwhelming for a lot of people to deal with so many parameters that they require adjusting to get a better glucose level. Another progress is organs on chips or cell on chips. A lot of cells that were happening that people grew in cell cultures and mediums, and they made discoveries, they did metabolomic studies, other studies, molecular biology studies, they always were intact. So always the question was, well, it's right you say that these changes happen in Petri dish, but would the same thing apply in real life? Because you miss that complexity. You don't have the same complexity as a real life cells within an organism has. So they managed to miniaturize this combination of different organs and cells and how they interact and evolve via this organs on chip. So metabolomics on chip is also now a fascinating way to study all these changes between different cells via different organs that you can continuously feed through a system of making it a continuous growth and changes and track back what comes out of these cells. So sort of exometabolome, endometabolome. This is also being used. So the exo being the metabolite that is in the environment, in the cells, in urine, in blood, whatever is excreted, what is the cell comes out of the cell. And the endometabolome would be what you studied with it inside the cell. So therefore you need to extract these metabolites out of the cell and use to be able to look at them and see what changes there are. Obviously, I'm not going through details, and each one of these fields, each area has some complexity, and many people are working, and there are a lot more. Each one of it is a subject that we want to be discussed, and there are far more experts out there who work on this, more know about these things to find a mechanistic view to talk about these things. I'm trying to just give an overview of how things are. So how many are there? Metabolites or metabolome. There's been hotly debated discussions for many years about, so how many metabolites are we talking about? Maybe we have by more and sequencing machine and my changes that happens in the past 10-20 years, we have more clear of how many genomes and genes are, although these changes in the same area is changing rapidly. And likewise, understanding of transcriptome levels are growing as well as proteome. So there's quite a number of these genes and transform metabolome are known, and the number of unknown is reducing in those sciences. However, the metabolome, it comes kind of contrary. It's that the number of known metabolome that we know of, or metabolites, it's a lot smaller than what we believe or perceive to be unknown. This might tend to be not true the case. The same thing that was the case for genomics, and people described that, okay, humans must have hundreds of thousands of genes, and then real numbers are far more lower than expectation of many people. But we know that certain classes, certain groups, for example, plants, have far more chemical diversity than, say, mammalians, because they need to have a fixed space in space, so they need to cater for everything without moving to be able to grow, to defend, to cope with the environment, while mammals or other animals will run for shelter, or when their food is low, they just migrate, while plants can't do the same thing, so they need to have more chemical mechanism to defend, to communicate. Therefore, the idea of compounds, the chemical compound landscape is far more complex and diverse in plant metabolome than, say, humans. Similar thing for organism, a microorganism is far more complex because they also communicate. They have their own macrocosm of a microorganism in a complex, in synergism that creates far more complexity in terms of number of metabolites and interactions that they do to cope with the environment that they are bound to. So this doesn't make the job of a metabolomics, a metabolite person, someone who's working metabolomics easy, because you got diversity, this chemical range, and this I will show in later slides. There are quite a number of degree of changes, dynamic changes between metabolites in all aspects. In terms of spire sizes, for example, there are 2 orders of magnitudes. In terms of concentration, there are also far more changes and magnitudes of dynamic ranges changes. I'll show that in the next slides. For example, I quite like this slide by Andrew Lane, and then he described me about metabolites. So how do I know how much of this metabolite are there in cells? How do I extract them? What are the expectation numbers? So I borrowed these slides from him. I hope he doesn't mind. But this is sort of an average human cell that is about 0.2 to 20 picoliter volume. About 30% of that is the biomass of it. This is what you burn and left, which about 20% being the proteome, something about 6 picogram of DNA. And out of that, on average, 10% of that volume in a cell is a metabolite. So be able to have enough sufficient volume of material to be able to do analysis, depending on technology, varies quite a lot. So you might need quite a large number of cells to be able to grow enough material of biomass to extract enough metabolites to be sufficiently used for the technology. I will again show a bit of more about how different technology in terms of sensitivity, because not all instruments are able to take all metabolite at different concentration level. Some are very easy. Those are in millimolar range, are usually very abundant metabolite, TCA cycle, Krebs cycle. Other cycles are more effective in terms of energy maintaining the cell. So they are highly concentrated. And as we go down, vast majority live in micromolar range, that it can be detected a lot easier by mass spectrometry than, say, by NMR as an analytical technique. And then you get to very, very lower range of nanomolar, picomolar range that you require very, very sensitive instruments to be able to pick that up. And this gets very close to analytical noise that I'll describe. Again, there's a degree of differences. So these rules, these numbers, do not apply to all type of cells, or do not apply to all type of variety of cells, whether mammalian or plant. It has so many factors that affect the number of mitochondria, defines how much metabolically active in terms of energy efficiency. So therefore, some metabolite TCA cycle might be higher in those with higher number of mitochondria than others. And this compartmentalization also is a key. And another area which is very interesting in metabolomics, there are very few labs doing it, is in single cell metabolism. There's a ETH group in Switzerland, which is starting, and one of the very first groups that are starting to work on single cell metabolism, which is of interest to a lot of people. But it's not as mainstream as other technologies in metabolomics. Back to this dynamic range. So what I meant by dynamic range are sort of depicted in this pyramid graph. So these are different elements of changes in the range of metabolome. For example, I already mentioned that size goes by 2 orders of magnitude. Usually metabolite is something that considered metabolite. These numbers varies depending on research. Other people between 1,000, 1,500 Dalton, anything lower would be considered metabolite, but there is a cutoff. And this cutoff could be about 100, 150. So some smaller molecules, like water molecules, like oxygen, or some others are not necessarily considered, for example, being metabolites, or something in between, or peptides these are, again, very philosophical, and if you talk to different people, they might have different ideas about of these numbers and what is the cutoff range. Again, there are differences in terms of chemical properties. Some metabolites are more hydrophobic, some are more polar, some are more volatile, and there's a whole degree of range in pKa and in terms of pH that they can cope with. And even analytical techniques are kind of highlighted here in terms of sensitivity. So being at the top of the pyramid is where you are mostly a larger molecules and highly concentrated one. And the bottom of the pyramid is where you get to lower concentrations and different range. So in terms of that, NMR is the least sensitive of the equipment, and type of mass spectrometry based on separation of chromatography or separation gas chromatography or liquid chromatography are more sensitive instruments. And then when you go to custom assays, you can actually pick very few metabolites. But then again, number of metabolites that you can detect changes a lot. So NMR tend to find or look for higher concentrated, therefore finds a lower number of metabolites, maybe in hundreds, while the same number goes up. About 300 to 400 metabolites you can usually find by GC, and again numbers go up to thousands of metabolites by LCMS. But then finding and identifying what they are is also another challenge, because the structural change happens to vary quite a lot. Something simple as lipids, so these are triacylglycerides, essentially 3 fatty acids bound to a common chain that can have and they're depending on presence, absence of double bond and length of these chains, you can have numerous species arising for it. So if you calculate it based on 40 common acid and 3 fatty acid per triacylglyceride, you can calculate that there are about 64,000 different species of them exist. And knowing and classifying them is quite hard. And identifying it is even more difficult to be able to do it, but there are progress. Slowly, more and more people are able to identify these things. So now more a lab-based practical view. How do we go by this? There's a several extractions method that you can apply. You collect the cells from the environment, hopefully with a very nice study design right from the beginning, in consultation with a statistician or someone knows about sampling. You collect the samples. Then you have to know what type of samples are. There are numerous ways that you can extract metabolite from the samples. Idea being very chemistry and chemical. You go by a method, you disrupt the cell, you burst the cell, you silicate the cell, you make the metabolite out, you provide different ways of separating them, depends on preferred to be a soluble metabolite in the aqueous layer or more a lipid. And then you use an organic solvent or aqueous solvent to separate them and spin it down, leave a palette of cells and DNA in between. You extract the metabolite, and then you have this choice of analytical technique used, which again is a complex and by itself, a long talk itself, goes into the domain of analytical chemistry. What I'm trying to point out in the center before I go through the pipeline of this example is that the complexity, the nice thing and difficulty thing about metabolomics same time is that it sees in the cross-section of different sciences. So it requires some chemical knowledge. Beside the biology, you know about chemistry, how these chemical reactions go, and you know about analytical chemistry. You have to know about your biology, and what are these techniques and engineering of instruments are very, very variety of them are depending on some sort of engineering settings that these instruments have. So they can interact and extract certain metabolites, so there are physical, chemical properties. So you will end up with large data that could be, depending on sensitivity in an instrument, could be a huge data matrix. This is the data matrix example coming from same sample, put into mass spec, and at the end you generate a data matrix. Handling, analyzing data, also it more and more requires some informatic knowledge, statistical knowledge, to be able to use and interpret these things into a model, and then listen to the model and see what model is trying to do and try to validate this model, try different methods of modeling. And there are numerous ways of statistical and computational modeling that you can apply to a data matrix to be able to interpret the data. And ultimately, these things have to have some biological sense. Just putting into a model is not a biology, it's not understanding. It needs to be interpreted in the wider context of a biological network, a pathway. How these changes really translate in a pathway? Do they map all of them belong to the same pathway? Do they show a trend of change in certain pathways? And how can I verify and validate these things to be able to interpret the results? So as you can see, it's quite a complex by itself procedures. And this is, when you interpret it and integrate these things with other omics, it's become even more complex. But this is the way we going ahead, and hopefully the means and elements in computer science can help and provide more means of better handling this data, understanding and analyzing. And this is the area which I'm more interested in the data side. So the snapshot point of view, which I've sort of highlighted, is only one story of the whole picture. My analogy to this being a train network, right? So this is UK train network. And you can see different areas are more dense. So sort of like, say, London area, a lot of train tracks, train stations, and traffic is there. Maybe this is more a hub of a country because a lot of businesses going there, a lot of trades going on, a lot of activity going on. And the same thing in a cell. There are areas in a cell that are more active, so metabolically more important to keep this running. So there's a lot of redundancy in this network to run. And some areas, if you go far rural area, maybe there's only one track line goes there, and any now and then a train passes by. So if there's any blockage there, it's not expensive for cell or expensive for economy. Similar analogy is there. So if people who are trying to work with these networks and try to understand how traffic are to be able to cope, they would have snapshot point of view. This would have only give partial information. So this information is still useful, but not the full picture. If I just set up a snapshot to people and say, this is the traffic in this area, just go by it, it's an information, so I can tell there's a lot of people going in this station, but I cannot tell what exactly happening, where they're going, or how they're coming. I don't know if it's true at 12 o'clock or same patterns at 6 o'clock or different in 12 p.m., for example. So these are information that timelines bring to a snapshot. So what people do in controlling a network, they require more continuous systems and ways to be able to see, to track these changes real time, to be able to cope with it in case there was an accident, so they can diverse the train. So this is what tells us this is maybe is important. So when you're thinking of your metabolomic study, when you want to collaborate with a metabolomic study, these are things to factor in, whether my timelines or time points and changes that I see, does it translate to some event? I give some examples. For example, here is an effect from Diurnal Rhythm from measuring PPAR, gamma, PPAR, delta, as well as how they translate and change over time. So the x-axis is a timeline in a mouse from 24-hour timelines. And you have daylight activity and nighttime activities. And some of these hormones and changes in leptin varies quite a lot. As you can see, for example, in figure B, in the wild-type mice, there's a lot of activity happens in midnight. So most mice are metabolically active during the night, and they evolve to sleep, for example, a lot of them in the daytime. But we don't do our experiments in the daytime, in nighttime. We do it in the daytime. So we are collecting not something a true representative of activities in there. So the changes from reflect into brown fatty acid and brown fat tissue. It also reflects the changes in diurnal effect. So more and more people are now have to consider these things, factor that for their study design, for their experiments. And as you can see, these levels go up and down. Sometimes, or for example, from 25 to 100 by 4 folds over a 24-hour period. So if I collect some of my samples in the nighttime, a certain hour of days, and the other one in 2 hours later, I might find a change or discrimination. But it's not because it's changing, because the values of metabolites, hormones are changing. They're not fixed during the time. So this fluctuation is also important. We all have this fluctuation. I give more chemical examples. If you go to a doctor's or clinic and you measure certain metabolites, they usually give a range. Say these are top and below this range are your healthy levels. These are levels that we expect to be considered on population average to be a right number. Whether that you fit in that population average or you're an outlier, it's a different story. But if you fit in that population average, these are the value and fluctuation you get. Sometimes you might go high, sometimes you might go, some of them will be lower of that scale, some of them will be higher of that scale. It is when we we find these things, the important is this early diagnostic. And this is what a holy grail of biomarkers. And one of the areas that metabolomics can play a big role by providing these biomarkers for this fluctuation to be able to monitor and change over time. Because this is what happens usually, for example, for this case, if there is a change that you switch from a healthy state to disease state, usually the diagnostic happens quite late. So the earlier changes, early fluctuation goes unnoticed. And when you have higher level, that's when you develop symptoms. So example, going back to type 1, for example, or changes in diabetes, you will not see these changes till your glucose level is really high up. And usually this is a very dangerous point, because a lot of early diagnosed patient with type 1 diabetes, for example, they're in ketosis, meaning the levels are so high that they're very dangerously at the edge of going into shock. That's where diagnosed. Well, these levels didn't shoot up in one point, but it happened over time. So some techniques, some analytical technique, which we are never far away, is part of that a network of interrelating sciences in metabolomics, that you have element of biology, element of chemistry, element of informatics, and now we are in element of analytical chemistry, and we have all range of equipments. I'm not going to go into detail of these, what exactly these equipments are, but there are a whole plethora of them. They manipulate different properties of metabolite, whether there is elements of ionizing it, making it into an ion, and measuring a charge value. They will measure different metabolites while they ionize, and how fast they fly, how big, in sort of a tubes, how fast they go from one, or how big they are, how small they are. By manipulating these physical chemical properties, by magnetic properties, you can find different instruments, and different instruments have a variety of detecting different metabolites. These are also can be separation technique associated with this. So GC part, gas chromatography, or liquid chromatography is only a separation. And then for that, you might have hundreds or tens of different type of columns or chemistry or solvents that carry this over. So this complexity is quite complex. And usually what I would always think, and I hope some of you disagree, is that metabolomics is a teamwork. A lot of teamwork lab required, because not everyone has full knowledge of this analytical chemistry, data, or biology, but it's a combination of these things. So you need a good mass spectrometer to be able to conduct and understand this chemistry part of it, set up the equipment. These equipments analogy for me is as TV. A lot of TVs and manufacturers come with standards or broadcasting method that gives information. Essentially, you're looking at the same event. You're looking at the same information. But depends on your resolution, besides how good the picture look, it's the detailed information that you can pick up. Maybe if you go have an HD or SD, you will not pick up as information that a 4K can give you. Similar thing that happens in mass spectrometry. The translation, the equivalent of this information, which again, we are looking at the same information. So what I'm trying to say is that the event, the samples, the biology is the same phenomenon. It's just the information that you can get by equipment varies. And it doesn't mean one more higher sensitivity is necessarily better than lower sensitivity. It depends on the question or biological question you have. And that's the key. Just because you have a 4K HD UHD TV doesn't mean you can't enjoy the same TV or same music or same program you're watching in a lower resolution. So that's information separation from resolution and details of that is a key here I want to point out. And without not going to different exact translation of what these acronyms means in type of mass spectrometry, but one analogy to that is the mass accuracy. This is how good resolution a mass spectrometer can pick up. So something like linear ion trap, there are the older standard of HD TV. They're still very good. You can still do a lot with it. But the resolution of it, the separation is, for example, lower than if you go with an orbit trap, which can pick a lot more discriminated differences by 1 ppm differences in how it's calculated. And formula below, the expected mass and calculated mass and how ppm is calculated is below. the message is here that it's all really application dependent. This is a lot of questions and collaborations or other people come up with. So which instrument is good to go by? Really, the most important thing is what you're after. Yeah, and the more information, the more sensitive, the more complex the data are. So the file sizes, which I haven't put there in this slide, which I borrowed from somewhere, I can't remember, is that the complexity of the information also goes up as the more higher resolution you have. So something with FD-IC-MS, FD-ICRM-MS, the complexity of data and the size of the data is far more bigger than a linear ion trap or a triple quad equipment. Another advantage, disadvantage, to see if you're thinking of collaborating or with a metabolomics group, or you were thinking of what analytical techniques they offer, which one should I choose, or working on your PhD and want to know which one equipments that are available to me, how do I choose and which one go by. This is sort of a brief pros and cons that I want to summarize in what's the advantage or disadvantage. So unfortunately, I don't have time to go explain what NMR is, and I hope you can Google it or look it up and see. Nuclear magnetic resonance is a very nice technique. It relies on a property in all atoms called spin, and it's a magnetic manipulation of these spins that gives these resonances. It's sort of the same MRI imaging that you might get if you happen to do an MRI and they can see the changes in water molecules. They will portray, depending on density of the water molecules, you will see this image of your, for example, bones or brain or skull. In there is information about protons that are attached to metabolite or carbon atoms, depending on experiments, and you will be able to see how much information there is. So NMR, the nice thing about it is there's no separation. Sample preparation is quite easy. It's a non-destructive method. The same way you go inside the MRI magnet and come out, hopefully the same person, samples go inside a magnet and come out as they were. There is no chemical reactions that requires, there is no ionization, there is no heating elements that are required to do other than what's normally just making sure it's equilibrium required. However, it is quite fast. The disadvantage of it, and you measure a lot of metabolites simultaneously. The disadvantage is it's not as sensitive. So as it's the best identification, you can only find metabolite in a higher molar concentration or micromolar range. And then you won't be able to see very, very low concentration or low concentration metabolite. Also, there's a lot of overlapping between signals that you need to decipher and change. So compared to mass spectrometry, it's far less sensitive. Also, it does not work very well with organic layers. There's a lot of overlapping signals that doesn't help in deciphering. And mass spectrometry, on the other hand, this is sort of put together. This is just a combination of mass spectrometry, regardless of the type of chromatography and separation. For example, gas separation, GCMS, or liquid separation, chromatography, MS. Their sensitivity is far better than NMR. It's a lot cheaper as well to buy an equipment. It's more difficult to make the samples because some of them you need to make a chemical manipulation. For example, for GCMS, you need to prepare them to be able to cope with very, very high degree of temperatures and become a volatile, to become in the gas phase, to be able to travel to the column, reach a mass spec. Numbers you can detect also goes up. So maybe twice or 3 times more metabolite that you can detect compared to GCMS. And there's certain metabolites that can become more volatile and go through the gas phase are better equipped to see by GC. And if it doesn't go, not all metabolite, as I said before, the dynamic range, property, physical chemical properties, some of them cannot be easily into a volatile. So maybe a liquid is better. And you can do several separation based on a liquid chromatography coupled to a mass spectrometry, and the numbers goes up again to 2-fold or more in terms of number of metabolites that you can do. Every step is also introduce an error. The more analytical chemistry changes you do, you might also introduce an error in your measurements. So in a sense, it requires more taking care of sample preparation. again, a lot of steps take longer to analyze data compared to NMR. But then again, the range of metabolites that you get is far more dynamic. You can have a higher concentration and lower concentration, very low concentration metabolite. Again, back to the sensitivity of equipment, the very, very low concentration metabolites that you can pick up, which is not possible by other techniques other than mass spectrometry, for example, compared to NMR. Another growing area is in imaging. It is very, very fascinating. It's different ways of using a laser or chemical ionization to be able to use a tissue or an extracted tissue of the cell and bombard it by high energy ions or laser. And what it does, it evaporates the metabolite from the surface of the tissue into a mass spec. And that's what you will be able to detect these changes. But the advantage of these things, you have locational information of those metabolites. So while I'll extract the cells, I will not know exactly where the metabolite concentration were in the cytoplasm, or whether they're membrane bound, or whether they were in cytosol or mitochondria or other organs. I lose that information because I don't have that spatial information. It's all extracted and pulled out, what I have in total average. While the changes in that is making it possible for me to see in a tissue, part of the tissue or cells, organs that where are the most concentrated, and be able to map those and find the spatial information. Again, this is a very nice area and growing area of imaging. Again, it has its own challenges and the complexity. It produces very nice images, as this example from our Metaspace partner, EMBL, but it's not an easy, a straightforward method by itself. It requires a lot of work. It's coming more or less to the final slides. Again, application of metabolomics. It's quite far and diverse. A lot of it, maybe I emphasize more on clinics side. That was a long time ago, more background information that I collected. And things are happening quite a lot in there in terms of, for example, iKnife, which if you Google it, you will find a lot of nice information about how these things could be taken to hospitals and clinic, and while they're cutting a tissue, and same time sucking the gas up into a mass spec and be able to differentiate whether where they're cutting is a healthy tissue or a tumor tissue. So with that, so this helps a lot in surgeons, for example, to be able to make cutoff. But it's not just in medicine. It changes in the environment, agriculture. Plant is even larger group of people doing metabolomics in plant sciences than in medical sciences. In environment, there's a growing area in environment, in oceans, and sort of you can see that wherever there's a huge textbook, there's a plenty of them on sort of area specific to microorganism, microbiome, plant. That means a lot of work, as well as nutrition. So nutriomics is also changing their words. A lot of people trying to create a new definition and keywords of omicking an existing nutrition to nutriomic, and working more focused on area and aspect of metabolism. So with that, just finishing on what we do at EBI, we actually, to say, because of it started, we are on the end of the data. Again, this is more towards metabolomics people, and probably most of them hopefully are aware of our services that we have at EBI, is we house this data. We capture metabolomics data, but people want to share and make it available to the community in a way that they can look at the data. As we know, publication might not be enough to reproduce the result. You need to be able to share this data. And sharing data is not as easy as providing a file with some link, because then there's hundreds more questions of, so what are these files? How can I use it? What are the tools? So we try to capture this information, as well as putting more structure in terms of informatics and tools that can be used more easy with people with not access to bioinformatician, to be able to launch tools, to be able to use tools in an integrated environment, to be able to look at, to analyze data, to share this information. As I said, a lot of more data, instruments are getting better in producing in data, making data. the bottlenecks always comes down to how we handle data, how we use the data, and how to analyze the data. And this is sort of what EBI is doing for all biological sciences. So metabolites and small molecules only is a very, very small part of a very, very large organization that handles information about proteome, genome, and all aspects of biological sciences. So we are a very small fish in a big, big, big pond of EBI. I put some references, so these slides will be available at some points that if you want to look and read more about extraction method or data analysis or visualization, things that I haven't covered, things about differences between data processing, data analyzing, using mass spec. And there's a numerous place that you can go. We got some online tutorials, whether they come from Metabolomics Society or via EBI training. Essentially, it's another introductory to the same thing, and there are areas that you can go up and look up if you want to find more about metabolomics. This is sort of a snapshot of a similar way of describing the basics of metabolomics in an online tutorial with you can go in different stages. Maybe it takes about an hour to go and provides different angles to what I already mentioned about metabolomics, that you can do it on your own time. So I guess this is where I finish my slide. I hope it was useful. Again, these are complex. If you have any questions, don't ask. It's a very collaborative. Not everyone has all the answers. There are forums, there are places, there are a lot of resources that you can reach to many people to be able to help you and guide you. And Metabolomics Society is another place. So I guess this is where I finish. Okay, thank you.
So thanks again for joining us, and we hope you have a pleasant evening.